Hello everyone, my name is Faiz Khan Sahil and today we're gonna learn about plant nutrition, okay? So, all living organisms require various substances to survive, right? These substances are useful for the body and the process of taking these substances is also called feeding or nutrition, okay? So the question might be why we are taking these substances, right? These substances are taken to create new body parts or cell division or to repair cells or damaged cells, right? Or to generate energy, right? Glucose is broken down to produce ATP, right? Glucose is broken down to produce ATP, right? ATP is simply a battery for a cell. Like you have battery in your car, in your phone, right? ATP is the battery for the cells. Animal and fungi cannot make their own food, right? Instead, they rely on the organic substances originated by plants actually. Even when there are some animals, right? Lion, cheetahs, tigers, they eat other animal. But if you trace back the nutrient, all of them actually comes from the plant itself. So the green plants actually make their own food by using carbon dioxide, and you hope you know this thing, carbon dioxide, water and air. The process is called photosynthesis, right? We're gonna learn in depth about plant nutrition, the photosynthesis and also several limiting factors, right? And disclaimer, I'm very new to the YouTube thing, so I'm a bit nervous. So stay with me and I hope you'll get a lot of values from these videos. By the way, I'm in my 4th year. I'm currently doing microbiology and genetic engineering, and my plan is to teach you guys about O levels and A levels biology. However, let's start. So stay with me, okay? This is think it as a live classes, okay? I'm a bit nervous, so stay with me. So the plant produce complex organic substances, okay, right? What is a product of photosynthesis? We know the main product of photosynthesis is glucose, right? Glucose is simple sugar, right? It is just a simple sugar, right? These simple sugar join together to form carbohydrate. They also form lipid, protein and vitamins also, right? So what is photosynthesis? Photosynthesis is a process by which green plants make their own food using energy from the sunlight. For photosynthesis to occur, carbon dioxide is needed, water is needed, and over here, sunlight is needed. Without sunlight, we cannot actually carry out photosynthesis. We're gonna learn about an experiment where we shows that light is necessary for photosynthesis. For example, over here, carbon dioxide and water from sunlight produce glucose and oxygen, right? And this whole thing take place inside chloroplast. What is the function? Inside the chloroplast, we have green pigment called chlorophyll. Okay, chlorophyll give the plant leaf its green color. Okay, this is interesting. So why the chlorophyll is green? Why any substance has color? For example, in the leaf, let's draw a leaf. As you can see, leaves are actually green in color, right? Why they are green? The question should be why leaves are green. This is because the leaf actually absorbs all the wavelength of other colors except the green. The leaf actually reflect. It actually reflect green light, okay? Green wavelength light. That's why the whole color is actually green. For example, this background is actually gray, right? You can see it. I don't know about the camera, but it is gray. So it absorbs all the wavelength of light except 2 or 3 colors, which combinedly make up the gray color, right? So moving on to our biological sector, you will learn in depth in physics. So photosynthesis occurs in chloroplast of plant cell. It is essential for life on earth. It provides food for the plant and release oxygen into atmosphere, which is used by the animal and human. Without oxygen, you guys will die. Okay, so the plant is essential for us human to survive. Even the glucose made during the photosynthesis is used to make various substances. First, it is mostly converted into starch, which is used as a storage form, right? And interestingly, cellulose is also made up of glucose. There are two type of glucose. One is alpha glucose, and another is beta glucose. Okay, you will learn this thing in depth in A level, but consider two type of glucose. Okay, so beta glucose, what is the function of beta glucose? So the main function of beta glucose, they form this structure. Oh my god, this structure is so bad. So they form a ring. Okay, they form a ring of glucose, beta glucose, to form cellulose, right? So look, alpha glucose is used by the cell for other purposes. Beta glucose is used by the cell to manufacture cellulose, right? And oxygen is needed for us for respiration. Now let's look at the uses and storage of carbohydrate in plant. So the glucose is converted into starch, which act as a reservoir in plant cell, and the glucose is also converted into cellulose, which build up the cell wall, which is rigid and strong in structure, right? It is used in respiration, where glucose is broken down to release energy, and the energy, as we described, is stored in ATP, adenosine triphosphate. It is converted also into sucrose and transported through phloem to other part of the plant. It also used to make lipid, long-term storage facilities, right? It also used to make amino acid, right? By combining minerals like nitrate to produce amino acids. Now we'll investigate the need of chlorophyll. Okay, glucose is produced in photosynthesis. It is quickly converted into starch. So by actually testing starch, we can actually say if the chlorophyll actually actually absorb light energy. So the photosynthesis occur inside the chloroplast. Okay. Oh, in your confusion, inside this is, for example, the structure of chloroplast. Okay, chloroplast. Inside there it is chlorophyll over there. Okay. Now let's look at the starch test. So what is starch test? So starch test, we actually test is the photosynthesis actually occurring or not, right? So first you take a leaf, and we have a beaker, and we have water. We actually heat the leaf in boiling water. Okay, so which denature all the enzyme inside the leaf. It denature all the enzymes. Okay, then it is as you can see in the test tube, it is actually put it in ethanol, so it discolorize by removing chlorophyll. Okay, by removing chlorophyll. Okay, after decolorization of leaf is put it in cold water. Then we do is iodine test. If there is a presence of starch, iodine will form blue-black color. If there isn't, iodine wouldn't form blue-black color. As you can see, it forms blue-black color, right? So starch is present. So we can say we need chlorophyll to for photosynthesis. Okay. Now let's investigate the need for light. Okay. This experiment I have done when I was kid. I guess I was in class 7 or 8. So what in this experiment, what you do, suppose from the plant, you don't cut the plant. Okay, so you don't cut the plant. Okay, from a plant you will take a tape that is not transparent, so light cannot move through it. It is put it over here. Okay, so if light isn't needed for photosynthesis, if we do iodine test, the whole structure should be blue-black color, right? So we only cover this portion with the tape, and the rest of the portion are kept so that photosynthesis could occur. Okay, now what you do, we keep the plant for for example 48 hours or 24 hours in the sunlight, right? In a sunlight, 1 or 2 days in the sunlight, then we cut the leaf of our hair, right? And we test with iodine. You can see the structure actually turn blue black, so photosynthesis occur and starch actually formed. Okay, now let's demonstrate the need for carbon dioxide. As we all know, for photosynthesis to occur we need carbon dioxide, right? For example, this is a jar where we have sodium hydroxide. Sodium hydroxide, what it does, it's actually absorb carbon dioxide. Okay, so the plant doesn't get carbon dioxide. As a result, photosynthesis cannot occur. So that we put it in the light. So after that, when we test for the iodine test, this structure, this experiment in this A, we have no photosynthesis could be seen. Whereas at the B, as it is a control, right? It is a control. Water is a control. Use as control over here, as water cannot absorb any carbon dioxide. As a result, over here photosynthesis actually occur. So iodine test positive. Iodine test is positive over here. So it turn blue black in color. Over here it actually didn't turn anything because the test is actually negative, because no photosynthesis has been occur. So now we're gonna learn about factor that is affecting photosynthesis: light intensity, carbon dioxide concentration, temperature. Okay, so light intensity, let's look at this graph. In this graph we can see rate of photosynthesis and light intensity in the x-axis. Okay, so as you can see, as the light intensity increases, the rate of photosynthesis increases until this point. After that point, no matter how much light you put over here, the photosynthesis will plateau over here. Photosynthesis will go plateau over here. This is because other factors are acting as a limiting factor. For example, temperature, carbon dioxide concentration. Let's look at another graph. Similarly, we can see the effect of carbon dioxide. The same axis, the level of carbon dioxide, the rate of photosynthesis. As the carbon dioxide concentration increases, rate of photosynthesis increases until this point. We call it x, right? So over here it is acting as a limiting factor. Why is that? Because other factors are acting as a limiting factor, right? So no matter how much, as you can see, the carbon dioxide concentration keep increasing, but the rate of photosynthesis isn't increasing, as you can see. So light intensity and temperature over here is acting as a limiting factor. Now let's look at temperature. As you can see, as the temperature increases, the rate of photosynthesis increases until this point, right? After that, all of a sudden, in this graph you can see the rate of photosynthesis plateaued, right? It didn't turn to zero, right? But over here it does actually, because the enzymes are actually made of proteins. So as the temperature increases, okay, as the temperature increases for optimum temperature, the enzyme started to lose its structure. The protein started to lose its structure. As a result, the enzyme denature at this point. So there is no rate of photosynthesis. Okay, we are up to this for plant nutrition. If you have any question, please write that down in the comments section, and please do subscribe and stay with me. See you in the next class. Bye bye.